All right, thank you. Our presentation today is it's called TidyCDISC. It is an open source application to interactively create tables, figures, and patient profiles from ADaM data. So I'm going to give you a spoiler alert. We're going to give a demo, but we also put it online for you to demo it yourself. So I am going to, that link is actually wrong, but if I'm going to share my screen quickly, let me know when you can see it. Can people see my screen yet?
It's loading. I can see now.
All right. So if you go to bit.ly/TidyCDISC, it will load a demo for you to interact with as we give the demo as well. This is using the Fuse sample data. So we have a sample ADSL, ADVS, ADAE, and ADLB, and you can use the application as we talk about it to show that it's, it's real, and you can ask questions and kind of play with it and see if it's something that will work for you. So now I'm going to stop sharing my screen. So the agenda, I'm going to give a 10-minute sort of introduction and slides. My colleague Maya is going to give a live demo for about 10 minutes. And then for the remainder of the time, there'll be some question and answer. What is TidyCDISC? TidyCDISC is an open source R package designed to analyze ADaM data. When you think of the name TidyCDISC, it's a play on the Tidyverse, which is a very, very popular package to analyze data in R. It has three major features: a drag and drop table generator, what we call the population explorer, which is a graph generator, and then a patient profile viewer, what we call the individual explorer. So there's been many attempts at this to create some kind of interactive explorer for ADaM data, to create something that will automatically create tables. And there's always been this tension of how to make this work between making something super flexible and making it easy to use. So we can think of it as a spectrum between flexibility and ease of use. You could make something that was incredibly flexible. You could write custom code for every table, but it wouldn't be very reproducible. You'd have to make something new for every single trial, and it would be very time intensive. You can also, on the other end, write a SAS or R macro to create tables. And it's very reproducible, very easy to use, but you're limited to a certain type of data and a certain type of output. So what happens is that you write the SAS macro and you validate it and you go through all this work. And what happens is that then somebody says, well, that table is great, but I want a slightly different table. And you either say no, or you modify that macro to do so. And then eventually what happens is that the macro becomes so complicated that you get sort of the worst of both worlds. It's not very flexible, and it's not easy to use. So you end up in this sort of cycle, this bad cycle, where you try to make stuff easy to use or flexible, but it doesn't work. So I think TidyCDISC is a step in the right direction to breaking that. And how did we fix this? We use the language of the web. There's a package called Shiny in R that how it works is that it creates a web page with the programming language R behind it. And what it does is it lets you have the flexibility and the ease of use because it allows the user to use the language of the web, which everybody is familiar with, to select options. And then it translates that into R code that you can use to customize your app or your graph or your table. How does it handle all your data sets? So this is a common thing of like, well, how does it work for this data set or that data set or another data set? And when you're creating an application like this, you have to remember that all Atom data sets are basically just the basic data structure. It's just a bunch of params. So in this example, I have an ADSL, we'll say a lab data set, like something like creatinine and calcium, and then a questionnaire data set, which is param 3 and 4. What we do with this application is that we take the inputs and we stack them together, all the params in one basic data set, and then merge ADSL into it. So you can just have one data set that you can customize and operate with. And because we just treat every data set as sort of a collection of params, it allows us to generalize it to work for any sort of basic data structure data set. We want to take advantage of the tools that are already out there. So there's a very vibrant ecosystem in R that can do a lot of table generation and figure generation, so we want to take advantage of that. So how TidyCDISC works is that you have a data import module, which is based on the ASA pharmacokinetic data import module. And then we use 3 major R packages. The table generator is based on a package called GT that RStudio produces. Our graphs are based on a package called Plotly. And our patient profiles are based on a package called TidyCDISC. Because we're using those open source tools, it allows us to focus on creating the custom JavaScript and Shiny code that basically customizes how the user can create the table. And as these packages continue to be widely used, we can take advantage of it. So a package like GT will get more options, for example, creating RTF tables. And then once those are updated in the GT package, we just slip it right into our package. It's also built for modularity. I have a saying that my needs are not your needs. So we can build something and it will be great for us, but we want it to be something that other companies can use. And so we built it basically to be modularized and added to. So everything you see that my colleague is going to give in a minute, everything can be added. Everything can be added or created additional graphs. So for the table generator, it can do 4 things right now: ANOVA, change from baseline, means, and frequencies. But how the main program works is that it just calls in other programs. So you can easily add something like a Cox model or something like a Cox model or like a chi-square test or something like that. It's very easy to add. And we actually have documentation about how to add additional graphs, additional things to the patient profile, and additional parts of the table generator. Well, with that, I think it's time to give the demo. I'm going to give the floor to my colleague, my Gans. I would also say, if you want to try to follow along, hopefully that link worked, and you can see you can do it with her.
So much, Nate. Please let me know when you can see my screen. Is the screen still loading?
We can see it.
Great. All right. So as Nate touched upon, this is our app TidyCDISC, and I just want to reiterate again, you can play with this yourself at bit.ly/TidyCDISC. So the landing page here is an upload module where we just, for the sake of the demo have the CDISC pilot data, and you can just give your data a perfunctory glance, make sure that you've uploaded the correct SAS files that you like. And now we're going to dive into our three. So first blocks that you can, for instance, we can take diastolic blood pressure for the ADV file and pair it with the block, and based on some we can calculate some basic summary statistics in real time for the diastolic blood pressure. Now, these blocks were made to we like to use the analogy of Legos a lot, so you can just build on this table. So we can stack our blocks, and I can do something like grab, let's say, race from ADSL, and then pair them, and you'll see again in real time now
we hear your
our
voice.
data.
Maya, sorry for the interruption. Your voice is a little choppy.
Yeah. Sorry, I could try to speak slower.
That would be helpful. That would be helpful. Thank you.
Okay. All right. So I've demonstrated here the mean block dragged in the blood pressure and race dragged in with frequency. And you see in real time, we see the number of patients and those basic summary statistics. And as I said before, but it was choppy, so I'm just going to reiterate, you can stack on as many of these block combinations as you like. We can also group our data. So if I want to group it by treatment, in real time, we see that now, rather than having a single statistic calculated, it's calculated per group. We can also, just as a proof of concept for some statistics, I'm going to drag in systolic blood pressure and are at, you'll see here that this p-value hasn't on those groups that you selected. So as Nate said, we have these 4 stats blocks here, but made it very easy for both the users and developers to add to this toolbox. It's also very common to want to filter data. So we've included a module for filtering on each of our tabs. So I'll demonstrate that here, where this filtering module populates for each column in data column, and add a filter like this, setting the safety flag to yes. And you see that this filter has been applied, and it's appended to the table's subtitle so that we've added this filter to the data. So we can filter our data, we can stack these blocks together. Another feature I will show is we have a palette of tables. So I need to create a demography table. We've created this custom generated table for you. And you can also, we've made this easy for developers to add their own commonly used tables, because we foresee those being different in different sectors. And then lastly, we can add our own title and then output the table as either a CSV or HTML table. Also, before moving on, I want to show you that on each of these tabs, we've included a help page. So if you click on anywhere you find a work, you'll see that we have a pop-up display kind of reading what I already was kind of touching upon. All right, so beyond creating tables, we can also create graphs. So I want to first show a scatter plot where you have a numeric column by numeric column. So we've automatically pre-filtered your data to only include numerics. And if you select the column that doesn't change throughout the study, like age, then you'll be limited to that selection. But if we wanted to collect a time-dependent variable like diastolic blood pressure, then we need to select a week that we want to measure that at, and we see that the graph is populated. We're using Motley for this so that you can hover over your points and detect outliers and get your data with more rigor in that way. You can also group your plots by a categorical variable, like, let's say, sex, and you can even color your plots. Again, I'm going to pick treatment just to show you can have these two categorical elements. You can also create spaghetti plots, which, similar to the scatter plot, is a numeric on the y-axis, and then a time-dependent x-axis. We can pick the visits over the duration of the study. And again, you can hover in here to try to make sense of, and now you can see why it's called a spaghetti plot. All right. Lastly, a box plot with a categorical axis, similar to the other two plots. We can plot something like AST over the different treatment in the study, and then you can toggle adding or removing points from the graph to inspect. Once again, if you wanted to filter your data, just as I showed you on the other page, you can do that here as well, and filter your graph downstream. Our last tool is the Individual Explorer, which shows you a patient narrative throughout the history of the study. So once again, common theme, you can filter your data, but I'm just going to select a subject here. And you'll see that we get their basic demographic info. And this module also creates a timeline of the patient's narrative over the study. So if we click on the events we'd like to plot on the timeline, here I've plotted chem labs, and I'll plot adverse events. This is an interactive timeline that you can zoom in and out time on the x-axis and see these events over time. And you just, as another way of displaying the data, we also have it as a table here. You can toggle over to visits. So for that same patient, just as we looked at that spaghetti plot for all patients we can pick a lab data set, let's say, and look at a parameter over time just for this singular patient that we're looking at right now. And we can even add a line to the graph to look at line, and those narratives, overlay them onto the plot. So here we see these vertical lines are the adverse events. Now, for this specific patient, we have these different parameters that we can circulate through to see over the duration of the study. So rather than usually have this really cool downloading room, which will create a report of a graph similar to this one, but for each of those parameters. So it's kind of my brief overview of some of the things that you can do with TidyCDISC. And again, the link is bit.ly/TidyCDISC. And we encourage you to play with it and would love some of your feedback.
Thank you, Maya, can you hear me?
Yep.
Okay, so that's the demo. It's feel free to play with it. I have the professional plan on Shiny Apps.io, so feel free to use it and play with it. So here's the question: where's the code? And so it is open source. The issue is that we sort of got a little stuck with the lawyers. Basically, we're trying to figure out basically what license to use, and that's been it's been a little bit of a question back and forth with legal. So right now, if you want the code right now, you can email myself or Maya, and we can set you up with it. My goal is to get it on GitHub, Biogen's GitHub, at the end of the month. We just kind of ran out of time, and you can't really tell lawyers. Like, I have a conference, they don't care. So, but it is going to be open source. We have commitments to make that work. It's just, we're just held up a little bit in a bit of a license question. I would be remiss if I did not thank the TidyCDISC team. It was a tremendous amount of effort. Everyone on this team did an amazing amount of work, and it's so cool, and I'm so excited to be finally presenting it. Everybody here deserves a massive amount of credit. And here are our emails. Nate.Mockler@biogen.com. Maya is maya.gans@biogen.com. We want this to be a collaborative effort. So this only works if a bunch of people kind of come together, and we sort of made the first step. But I'm hopeful that we can create something that will hopefully break the cycle, because I know every company is sort of working on some initiative similar to this. So I'm hoping that if we publish ours, it can start the process of starting to collaborate, starting to work together, so that we can create this platform for atom data exploration that I think would be a great boon to the data exploration process for all the companies, if we can start to collaborate. And now I'm open to any questions if anybody has them.
We do have some questions that came through. Where the data stores when we upload data sets? What are the data protection measurements?
Okay, so that's a good question. So this is something that's on a Shiny application. You can either, if you deploy it locally to your computer, it stays on your computer. You can also deploy it to something called a Shiny server or an RStudio Connect server, and the data would be uploaded into RAM there, and then deleted when you close your application. I think when it comes to data protection, I think that's something everybody's IT sort of data infrastructure is different. So what we did is we created something that's completely neutral, and you just upload the data into the application, and you can run it locally, because I wanted it to be agnostic as to how it worked with your data. So that would be something where you would have to, I don't know if you have Shiny applications on your servers. If not, you can always run it locally, and it'll stay on your machine.
Thank you. The next question is, will the link be valid after today as well?
Yes, it will be valid up until I stop paying for Shiny Apps.io, and I think I'm going to be paying for it for a while, so that link should work for the foreseeable future. And if you have any questions or you want like a private demo, you just email me and we can set something up.
Thank you. Why TidyCDISC instead of JMP Clin?
Okay, so why not Jump Clinical? Good question. So Jump Clinical is, of course, a paid application. You have to pay for it. Also, I think there's a lot more potential with this. This takes advantage of the open source ecosystem. And you have so many, like R has so many R basically has every statistical test I could think of in there, along with some great data visualization packages and great table generation packages. That was always sort of the thing about R, is that it didn't really, until recently, have great table generation packages, but I think GT is going to be a game changer. If you look at it, It's basically proc report in R. And we've integrated all of those into it, so we can take advantage of that open source ecosystem into this application.
All right, next question. This was great work. Do you have any challenges with large ADLB dataset from trials? Are you able to use the outputs in CSRs?
Okay, so with large ADLBs, it will try to load it into RAM. So that's based on your computer and your server configuration. So it's based on sort of your RAM configuration. So it's limited by your computer, and that's the application. It's not limited by. So I would try something smaller for now if you're going to upload a smaller ADLB, but the concept is the same. I forgot the second part of the question. What was the second part?
Are you also able to use the outputs in CSRs?
Okay, so once again, using it in a CSR means that you're integrating this into your sort of clinical output validation system, and every company is different. So we once again wanted to create something sort of agnostic. Where I think we are right now is doing it sort of. When you think about creating CSRs, usually I always think about it as like you have to create like 5 tables before you create the table that you actually want. So for now, because that sort of data, that sort of output integration process can be complicated, I see this as like you can create those 5 first tables in seconds. And then once you find the table that you want, you can go through your SAS process or whatever. So I see it as complementary for now.
Thank you. We do have 2 more questions about the size limitation, which I believe you already answered. The next question that we have is this: the slides button doesn't work for me. How can we obtain the slides? So
slides will be available. Yeah, the link doesn't. I changed the link, the Shiny Apps.io link, because I'm doing a run through this. Someone said, "Why don't you just use Bitly?" And I was like, "Yeah, we should." So I changed it. But the new slides with the updated link should be available after this meeting. And if not, just email me and I'll send you the link.
Yeah, and the FUSE website should also have the save and slides available. The next question, I think this would be the last one. What scenarios are the output of this tool intended to be used in? Exploratory, internal meetings, submission?
So my goal would be all scenarios, but I think we need to start with exploratory for now, because you get into sort of like regulatory would be awesome, but that's a long way away because you have to worry about your sort of integration with your other systems. So every company has their own sort of system of how they create TFLs. Obviously, I don't know every system. So for now, if we could create something that is so good at exploratory, then this question is obviously, well, why can't we just use this for the next step? But we need to get the first, like that's like the last step. The first step is to create something that is good at exploratory, because once you get the exploratory stuff done, then you can start worrying about all your regulatory requirements.
All right. Thank you. Thank you, Nate. Thanks, Mike.